In this video, you'll learn whether scientists can literally bring extinct species like the woolly mammoth or Tasmanian tiger back to life. We'll explain the idea of de-extinction, creating animals similar to those long gone, and focus on high-profile projects like Colossal Biosciences' Mammoth and Thylacine efforts, and what experts say about them. National Geographic defines de-extinction as the process of creating an organism that is a member of an extinct species or is closely related to an extinct species. As we explore, remember, this isn't Jurassic Park. The goal is usually to engineer hybrid creatures using living relatives, not truly resurrect dinosaurs or Neanderthals. Extinction is the permanent loss of a species. Humans have driven many extinctions, triggering a biodiversity crisis. In response, some scientists ask, instead of only preserving what we have, can we revive what we've lost? Proponents of de-extinction argue that reintroducing certain species could help restore ecosystems that went out of balance. For example, National Geographic notes that adding back an apex predator like the thylacine could restore ecological balance in Tasmania by keeping overabundant prey in check. Similarly, hypothetical mammoth herds could convert today's Arctic tundra back into productive grasslands, potentially slowing permafrost thaw and climate change. In short, supporters see de-extinction as a high-tech form of conservation or ecosystem repair. Critics counter that it may distract from protecting existing life or fail to solve the root causes of extinction. Creating a de-extinct animal is enormously complex. In general, teams follow steps like these. Collect and sequence DNA. Scientists gather DNA fragments from museum specimens or fossils. If possible, they also sequence the genome of a close living relative. This gives a genetic blueprint. Edit genes. Using CRISPR and other tools, they edit the living relative's DNA, inserting genes or mutations from the extinct species. As Harvard geneticist George Church explains, we're de-extincting genes, not species. For instance, Colossal plans to change dozens of genes in Asian elephant DNA to confer cold-adapted mammoth traits. Create an embryo. Edited cells are grown into a viable embryo. In principle, biologists can synthesize large fragments of DNA and use them to engineer a cell with the extinct genome. Gestate and birth. The embryo must be carried to term. Options include transferring it into a surrogate mother of the close relative or using an artificial womb. For thylacines, researchers propose using the tiny fat-tailed dunnart as a surrogate or an artificial womb. Each step is speculative. So far, only one full de-extinction has succeeded. In 2009, scientists cloned an extinct wild goat, though the clone survived only minutes. Even cloning living endangered animals has only recently succeeded. A black-footed ferret was cloned in 2020. So the idea is rooted in real science, but creating a healthy breeding population from extinct DNA remains unproven. The woolly mammoth was an Ice Age elephant that went extinct roughly 4,000 years ago. Harvard geneticist George Church co-founded Colossal Biosciences to bring back a mammoth-like elephant by editing Asian elephant genes. Colossal has raised over $75 million for this goal. Their plan is to insert key cold weather adaptations, hair, fat, blood proteins, into elephant DNA so the hybrid can survive Arctic climates. Church sums up the approach. The goal is really a cold-resistant elephant that is fully interbreedable with the endangered Asian elephant. In other words, it wouldn't be a pure mammoth, but a living elephant with mammoth genes. Colossal's timeline is ambitious. Church believes their first hybrid calf is about 6 years away, with a self-sustaining herd decades off. CEO Ben Lamb emphasizes that most of the science had been solved. They just needed that funding and focus. Some scientists are excited by the broader benefits. Colossal and others hope revived mammoths or proxies could help combat climate change. The Zimov's Pleistocene Park project shows that large herbivores can cool permafrost by fostering grassland ecosystems. As conservation scientist Ryan Phelan says, de-extinction is a story about hope and being able to repair some of the damage that humans have caused. It's not nostalgia, it's really about increasing biodiversity. The Tasmanian tiger, Thylacine, was a striped marsupial predator native to Australia. It survived on Tasmania until the 1930s, when hunting and habitat loss wiped it out. The last captive Thylacine died in 1936. Today, it symbolizes loss and regret. Recently, a Texas startup teamed up with University of Melbourne geneticists, the Tigger Lab, to attempt Thylacine de-extinction. They have already sequenced the Thylacine genome from museum specimens and are developing the required tools. The approach parallels the Mammoth effort. They will edit the genome of a living marsupial to approximate the Thylacine's DNA. The Thylacine's closest living relative is the numbat, sharing about 95% of its genome. Researchers plan to use CRISPR to genetically engineer a numbat cell to turn it into a Thylacine genome, and then develop that cell into a living animal. In practice, some have also suggested using cells from a fat-tailed dunnart and gestating embryos in a surrogate dunnart or artificial womb. If all goes well, lead scientist Andrew Pask estimates that the first Thylacine joeys could be born in about 10 years. Yet enormous hurdles remain. The biggest is genetic distance. Unlike the Mammoth's elephant surrogate, the Thylacine has no closely related surrogate. Museum DNA is often degraded, making a complete genome hard to assemble. Prominent researchers caution that de-extinction may not truly resurrect the original animal. For example, National Geographic explorer Chris Helgen notes that the Thylacine's only feasible surrogate is a tiny marsupial, as genetically distant as a human is from a marmoset. In his view, the idea that we can bring back this carnivorous marsupial, is unrealistic. Technically, many breakthrough steps must succeed. Editing hundreds of genes, creating viable embryos, and incubating marsupials in artificial wombs. Even if one embryo is born, raising enough animals with diverse genetics for a healthy population would be daunting. So, can we revive the mammoth or Tasmanian tiger? In short, science is rapidly advancing, but success is far from guaranteed. Current projects like Colossal's aim to produce hybrid offspring within decades, but both technical and ethical challenges loom large. Even if a creature is born, it won't be the same as its extinct ancestor, and releasing it to the wild would raise new issues. What these ambitious efforts show is not only how powerful genetic technology is becoming, but also how fragile our ecosystems are. As one expert summarized, de-extinction forces us to confront our past mistakes and the urgent need to prevent new extinctions. In other words, even as we chase mammoths and tigers in the lab, the greatest lesson may be to cherish and protect the wildlife we still have.